And as you already introduced, the second presentation given by me and Sven Behnke on behalf of the other PIs of Core Project 1 is an update presentation of what we are doing and what we have achieved in the past years and months in Core Project 1. Speaking of Core Project 1, just to set the scene, hang on, it's not moving now here. So the background of the PhenoRob consortium as a whole is that we want to combine the plant phenotyping, robotics, and machine learning for an automated quantification and of plant traits and the general interpretation of this data. And the objective of our core project, of the Core Project 1, is that we are the measurement enthusiasts, and we want to create the time series of really high resolution 3D models based on real field data from real sensors in the field to be able to extract this novel phenotypic features which we can access by having a close look into the plant structure and function. The goal is not in the first instance to have an operational system already on the market, but to really improve our scientific understanding of the dynamic interplay between the structural and functional dynamics. So if we map this to the objectives of the PhenoRob cluster, our main contribution is to the objective 1 and 5. So again, we are developing a lot of methods and approaches, which is a scientific challenge by itself, but we also understand ourselves that in the interlink with the other researchers in PhenoRob, we provide and maintain the infrastructure and the data flow for the research. So this also, with this first step in the scientific understanding, we contribute to the general PhenoRob vision that we think that future automated detection and understanding of the dynamic is the baseline for knowledge-guided management and breeding. What I have said now, this was just the general overview. We can also visualize this, how we understand ourselves, the Core Project 1 being positioned within PhenoRob. And there, on the one hand, we have collaborations and links to Core Project 3 and 4. We in Core Project 1, we kind of have the easy thing. We measure the easy accessible part of plants, namely the above ground part, the shoot. And we are there talking to colleagues of CP3, which are going below ground soil and root measurements, which is technically from the measurement modes much more demanding. But of course, we want to have the information about the whole plant. And there are also a lot of collaborations together with CP4, the colleagues in CP4, which are developing the automatic acting, while we in CP1 are doing the automated sensing. If you think about data and data flow, there you see, we see ourselves that we have the link to colleagues and work in CP2, 5 and 6, where on the plant modeling CP2, we are providing data and listening to the needs, which is expressed by the plant modeling people, but also on the large scale processes, all our detailed understanding in CP1 on the 4D crop plants is then the basis for the larger scale processes, which is attempted in CP5 and CP6. So this, just for everybody to be on the same scheme again, this is what we want to talk today about. CP1, the measurement and the development project for new measurement modes. And what we want to give you today is an overview on our status, where we are at the moment. On the one hand, in our area where we develop and optimize approaches for precise 3D construction of plants. And we don't only want to do this once, but we want to do this in time, which gives us really then the 4D reconstruction of plants in high precision. A lot of the approaches which you will see come from ground-based vehicles. We have to be close with our sensor to the plants to achieve the high resolution, but it's not limited only to ground-based measurements, but they are normally close to the plant. In the second part of the talk, we will move a little bit away. A lot of the work is done with drones, but also other sensors where we move away from the plant, having higher throughput, higher coverage, where we move therefore to the really the field scale, and then we try to quantify plant structure and function from whole canopies, plots, and whole fields. And as a new things, because this was developed and emerged in the past year, I would say, is that we now use the different measurement modes, the different approaches. We combine this on the appropriate scale, and there we come really from the biological questions, like what do we want to know about the performance of the plants in the field, the dynamics of the plants in the field, and therefore we then combine the different measurement modes as they become available. So with this as an introduction and an overview of how we understand the link of the different projects in CP1, we will now expose you to updates and highlights which come in these different focus areas. And with this, I would like to hand over to Sven Behnke, who will start close to the plant with detailed structural reconstruction of the plant. And with this, Sven, I stop sharing and you can take over.
So I hope that worked. And yeah, here you see the ground robot that was developed for CP1 from the initial planning stages over the first iteration. Yeah, the iteration that was used last year for measuring in the field, but this is not really the end of the story. Yeah, so we every year collect some data, make experiences, and then use that to revise the platform and improve it over the winter for the next growing season. There is different sensors on this ground robot. One was mounted on top by Felix Esser, supervised by Heiner Kuhlmann. And the objective here is to measure entire plots while the vehicle is driving. Yeah, so this is a LiDAR-based sensor that can measure distances. And ideally, there would be an animation. Oops. Yeah, now it seems to work. Yeah, so that these distance measurements, they can be geo-referenced with RTK, yeah, so differential GPS, so that you know where each measurement is and can revisit it also like 1 week later. So this has been deployed on the Phenorob experimental field in Campus Klein-Altendorf and was used to measure different kinds of crops. You see here that there is quite a high resolution in these geometry measurements. And if you come back on different days, you can revisit the same plant and watch it grow. And there is ongoing work on analyzing these geometry measurements, for example, to estimate the area of the leaves or the angle of the leaves of the different species. What is currently ongoing is that this single LiDAR will be replaced by 2 laser sensors that can see from different angles, so there will be less occlusion, and it will be complemented also by color and multispectral cameras. Furthermore, this ground robot is equipped with 14 at this point high-resolution cameras. So every camera can capture 45 megapixels. And the idea is to have really high-fidelity images from different perspectives on the plants, so that we can reconstruct the structure of the plant but also its geometry with millimeter resolution, and then also texture with maybe 0.1 millimeter resolution. And yeah, we went to the field on multiple weeks to see how these plants grow. And now processing this data is a major challenge for many reasons. And the first step in processing this data is to do structure from motion. Multi-view stereo methods like ColMap, they allow us to estimate the intrinsic camera parameters, but also estimate where these cameras are. So this is not really fixed because the vehicle is not really rigid. So when it drives over the field, then the calibration is not constant. But we can process the data and estimate all these parameters, which gives us a semi-dense 3D point cloud with color. But yeah, we can also use these multi-view stereo methods to more densely reconstruct depths. So this is like RGBD images from different views. And what you see here is a visualization that combines two meshes of these RGBD views, which are from a similar perspective as the viewer pose during these animations. And it's clear that there is a high level of detail with regards to yeah, the color and texture, but it's also clear that these reconstructions are not very complete. So there are some parts missing, as you see here. And this is due to the requirement that multiple cameras must see a surface element, and there is occlusion and so on, so that yeah, that is actually a challenge. To address this challenge, my student Alex is looking at neural radiance field-based methods. So they have the objective to render images that look like the recorded images. And in addition to that, Alex used the sparse steps coming from structure from motion, multi-view stereo, as an additional supervisory signal. So he also says that at the points where we think we know the depths, that this depth image should look like what we think it should look like. And this allows to give us a quite complete view on the plant, which is at this point a bit less detailed than the actual texture that we have from the individual images, but it's already yeah, quite complete, as you see here. In related to open call to CP1, I supervise also an open call 1 project where we essentially follow the same objective, but with a different hardware platform, namely a very small and lightweight, inexpensive, and not dangerous flying robot. So even before the project started in October, we made some measurements in the field, and even though this is cheap consumer hardware, there is a 12 megapixel camera where we can make high resolution pictures from different angles onto the plant. And yeah, here you see an example where we have been circling around the plant in 5 different elevations, made about 200 images, and used this instant neural graphic primitives method to obtain like the visualization and a depth rendering of the plant from novel viewpoints. Yeah, so I think that this is quite interesting, but it has also some limitations because here we cannot record the plant at the same time from different angles. This means if the plant moves a little bit, like here visible in this leaf, then yeah, there might be a little bit of an issue here, and we have to address this movement when we want to process this data in a consistent way. In the PhD project supervised by Maren, Tobias Zenker is looking at moving the camera such that one can get a more complete view of the scene. And so far, this has not been integrated to the ground robot, but it's done in the greenhouse with this mobile robot developed by the group of Chris McCool. And there is a neural network for image-based fruit detection. And the measurements are aggregated in 3D so that a view planner can answer the question, where should I move my camera next to get a more complete view? So that is answered in terms of information gain. And Tobias is working ongoing on this graph-based view motion planner with graph neural networks on representing the shape of fruits using geometric primitives like super ellipsoids here. And we plan to add a camera setup with 5 20-megapixel cameras and an arm for this growing season to the ground robot of CD1. Oh-Hoon Kwon, supervised by Eduard Zell, works on high-resolution stereo matching. So commercial RGBD cameras have limitations, as you see here. And we do have high-resolution cameras, but we need specific methods to reconstruct depths from these. So Oh-Hoon developed a method that is faster and needs less memory than competing methods, and applied this to measurements from the greenhouse of these peppers. And you see here that the depth reconstruction is somewhat more consistent than the commercial cameras. Moritz, who is also supervised by Eduard, looks at photometric stereo. So the idea here is to actively light surfaces from different directions, and then one can estimate directly the surface normals. So detailed geometry can be obtained from the differences of the different lighting directions. So that is very good. But it doesn't give absolute depth. So this surface normals, they need to be integrated, and multiple cameras help to do so. So to some degree, also the triangulation depths from multiple cameras is needed to address depth jumps over the edges of leaves. And the experiments you see here have been made in the dome developed by the group of Reinhard Klein, computer graphics in the computer science department here, where 2 cameras were used and only half of the LED lights, so that the true color of these surfaces and the geometry and surface normals can be reconstructed from this artificial plant. There was also an effort to publish data that has more or less the properties that I'm aiming for. But this data has not been recorded on the field, but in the lab with a handheld precise scanner. So the plant was scanned from close proximity from different direction, which gives a very good geometry description in terms of the point cloud. This was done for multiple days over the growing period. And then we get data like this, where we can apply machine learning methods, for example, for segmenting the individual leaves, as done by my student Alex with his lattice net approach, But there are also several other interesting use cases that already use this data, for example, for associating the leaves over time and modeling the growth of these leaves. Federico, who graduated from Cyril Stachner's group and is working now as a postdoc there, is looking at shape completion. Yeah, so the idea here is twofold. One is that he uses templates that are registered using a special transport distance to 3D point clouds obtained from a flying robot, in this case of sugar beet. And the color here indicates deviations that still remain. Yeah? So sort of the error that is minimized. But the second approach that he developed uses the good data of a particular category of shape, in this case, strawberries. And he learns a model of these shapes, which can be used then to complete data. So usually if we measure, we have only a partial view from one side onto the fruit. But this method can guess how the fruit looks like at the side that was not measured and register the template. Another work of Federico is towards less annotation of data for semantic segmentation in this case. Yeah, so if you have a certain domain and you have annotations, you can train here a deep neural network, for example, and then you obtain good results. But often you may have other images that you want to classify, and you don't want to label these other images. So the approach is here that generative adversarial neural network is used to make these source domain images look more like the target domain. Yeah, so it's kind of style transfer. So it makes this look more like this, but it has the same labels. So we can train on these adapted images. And if you annotate and train the target domain, you get, of course, good results. If you train on the source domain that is different from the target domain and test there, the results are awful. But if you make these target domain images look like, so the source domain images look like the target domain trained there, then you are almost as good as training on the target domain. So you don't need to annotate here, but you can transfer the labels to this similar domain. In a similar direction goes the work of Gianmarco, also supervised by Cyril. Here, self-supervised pre-training is used to learn representations which are then useful for downstream target tasks like semantic segmentation here. And it's clear, the more labels you use, the better your results. But also, if you use that pre-training, you get good results even with few labels. Similar, if you have a different task like leaf instance segmentation and do the pre-training, then it works better than if you don't do it, or if you do pre-training on a general image domain from the internet. And in addition to this self-supervised pre-training, also data augmentation techniques are used here, which exploit certain knowledge about this agricultural domain. So what are the typical variations in the data which need to be addressed in order to transfer the labels? With that, I stop here and we switch back to Uwe.
Yep, and perfect. So I think you can, I hope you can see my slide again. So now, thanks Sven, after the overview on the close range measurements, I would like now to move a little bit further away from the plant. And the first project is coming from the PhD work of Ansgar Dreyer, supervised by Lasse Klingbeil. And Ansgar has mastered in the past months and years the challenge to mount a high resolution penetrating LiDAR system on a larger drone. And in the last vegetation period, he was able with this system to really record an unprecedented high resolution 3D model of selected plots in our fields at Campus Klein-Altendorf. You see there an experimental small movie showing the growth of winter wheat during the vegetation period of 2021. And so Ansgar, after he has now the measurement system in hand, he has also done a detailed analysis on the precision and accuracy which is able to be achieved in the combination of a wobbly drone, which nevertheless has a high resolution GPS IMU system and this laser scanner. After this, he has done the next step and has combined this base map of the LiDAR 3D map, which develops in time, with other measurement modes, and he has combined this with RGB imagery and also with a ground-based data. And in the following, I would like to show you a visual what he can achieve. And the important thing to point out there is that there's no empirical registration of the different data needed there, but as the data are perfectly or with a high accuracy registered in space, in the 3D space, the different measurement modes really fall together in this universal coordinates. And we can start, for example, with a classical UAB RGB image. There you still see our ground robot. If this, however, moves away, Ansgar could overlay multispectral data on the same plot. He registered it, or he overlaid it with a UAV laser scanning system, which I mentioned before, or also with a ground-based laser scanning system. And what you can see there also visually, but there's also a quantitative analysis behind that. What you can see there visually is that the different measurement modes on different spatial resolution actually fall in place automatically. And this was one of the great technical achievements which we wanted to achieve. If you remember to the very beginning, if we want to go in an automated workflow where the different measurements can be automized, such a step was a technical prerequisite. And I think I could show you some visible inspection that I think we are pretty close on this step technically now. Having presented this, I would like to move to the second PhD project, which is the PhD project from Erikle. He works in my group, and ECHO is using a 10-channel multispectral camera system, also mounted on a drone. And you have seen the data before already in the previous slide. And what ECHO wants to achieve is that he uses this 10 color channels in a so-called radiative transfer inversion, RTM, radiative transfer model inversion, where he uses a combination of a leaf radiative transfer and a canopy radiative transfer model. He used the ProSail model, and he inverts this model, feeds in the 10-channel reflectance and radiance data from the drone, and by inverting this, he can then, without any empirical approximation, but with a physical-based approach, he can estimate and quantify relevant plant traits. One technical challenge which he had to solve before going there is the question of spatial resolution. Those radiative transfer models were originally developed from coarser scale images, from satellite images or airborne images, where you have a mixed pixel, a mixed signal from leaf level processes and canopy level processes. And ECHO, for the first time, used this high resolution, spatially high resolution data to adapt and test this radiative transfer model inversion. And in the next slide, I show you some images where he used this high resolution data, for example, to retrieve LEI, leaf area index, leaf chlorophyll content, or canopy chlorophyll content. And then with this, he went into an analysis on the payoff between spatial scale, spatial resolution, and error in the single pixels. And at the end, he could show that by using this high resolution data, as I show them here, you have a slightly higher accuracy and precision for these traits which he has tested so far, but also on the coarser scale traits, on the coarser scale resolution, nevertheless, we do not lose too much. And with this, he has established the basis of the optimal trait for this radiative transfer inversion if you want to look, for example, on leaf traits. This in the case is leaf chlorophyll content. There it also makes sense that you more use data where you see the single leaf, or if you look on canopy traits, if you, for example, want to retrieve LEI. So this is the status where ECHO is. So the method is established also based on multi-temporal data from last year. So it's also stable across different illumination conditions. It's stable across the growth stages which we experienced during the vegetation period. And now ECHO is moving forward and takes the next step. So, and the rationale behind that is if you have a really physical-based radiative transfer model, which you can assume that this is correct. So if you have this physical-based radiative transfer model, then you can also retrieve and invert traits which are not visible with the classical empirical methods, like, for example, leaf angle distribution, which is an interplay between function and structure. So we are just in the interlink between leaf traits, canopy traits, and leaf angle distribution is something which now we try to, or ECHO will try to get as one visible or invisible trait so far, inaccessible trait so far in the upcoming vegetation period. There, you, for example, see that we still have some influence of illumination conditions in our first tests, but we are optimistic that he can solve this in the upcoming vegetation period. With this, I have given you the overview of the two core projects which move a little bit further away from CP1. But now I would like to highlight a few other projects which we ramped up in the past year, where we really come from the biological question, like where we want to use our measurement modes, which are on an advanced stage of development, really for biological questions. And one is a project which is currently the PhD project of Julie Krämer, supervised by me and Thomas Döring. So you already see the link between different core projects. And we want to see how really actually, or what is the basis of the beneficial phenomenon in mixed cropping systems, which is well established. So it's well established that normally in mixed cropping systems, you have a higher yield, you have a higher resilience, and there are also mechanisms known behind. We know that the sharing of resources below ground is one of the important factors, but also on the other hand, the sharing of the light intensity above ground is a second important factor, which is underlying or the reason for this beneficial mixture effect. And Julie looks on the above ground part, and she uses two measurement modes, namely reflectance measurements and fluorescence measurements, to unravel the two underlying reasons. So she wants to know what is really structurally related, because light is used more efficiently and absorbed more efficiently in a diverse canopy, and what is related to the upregulation of photosynthetic efficiency on the leaf level. And by looking into the leaf level, she uses the fluorescence signal. So canopy structure, leaf level, function, interplay, and these two parameters Julie wants to unravel quantitatively, non-invasively, because she wants to do this over the course of the whole vegetation period. Some first results. It's a PhD project which started a little bit later, but nevertheless, we are in the moment at the point where the methods are established. So we know what traits have to be measured on the leaf, what traits have to be measured on top of the canopy. And we are also quick enough to carry our instruments around either manually or by machinery to achieve the needed throughput. One first result from this is that we, for example, look on the top of canopy emission of the fluorescence signal from the monoculture bean and wheat. We have two contrasting partners, high fluorescence, low fluorescence in bean, and also 3 wheat varieties with contrasting fluorescence emissions. That's how we choose them. And if you look at the mixture, it's a little bit complex, but believe me, we think that we see the forward propagation of the different partners in the mixture. We are pretty confident that we also see this in our mixture setup. So and this is the start that Julie will continue working in the 2022 vegetation period with a similar experimental setup. Second new project, which is even newer than the one from Julie, is a project which we just started together with Anneliese Mason and supervised also by me. The PhD student behind there is Theresa Sandmann, who has spent the past month to understand and to link the requirements of the breeding background coming from Anneliese with our possibilities to measure breeding relevant traits for brassica breeding, and to match this with the potential of the measurement modes which we have in hand. And I want to refer to the talk which was given in the same seminar by Anneliese Mason just recently. It's about brassica. And brassica, as you may remember or may recall, is a really important economic plants for our food production system. A lot of the plants which we eat are brassica species, and they are mainly derived of 3 ancestors: Brassica nabus, Brassica juncea, and Brassica carinata. And the commercially used plants are then hybrids of these. And for the upcoming vegetation period, we have now, or Theresa has selected a good sub-selection of different brassica accessions, which we will plant in the field. And we will look on one trait, namely the drought resilience in these traits, because we are convinced that early detection of drought is something which benefits from a close look at the detailed quantification of the brassica. So this is a new project now ramping up in the vegetation period 2022. And having said this, I do not want to miss to highlight 3 other projects which are always called partner projects. So there are 3 PhD projects which are supervised by Heiner Kuhlmann, and they are also linked to CP1, either methodologically or from the interpretation of the biological data. The first one is the project of Lina Zabava, and she again is using high resolution point cloud to transfer this in an automated pipeline in a surface mesh. And then the contextual link is, again, she is interested in leaf angle distribution, which is the distribution of the leaves in space, which changes, and this is the example there also, which is a variable which changes depending of the plant or the environmental conditions you're looking at. Second partner project to mention is from Laura Zabava. They're actually really sisters, so same back name, and Laura is interested in wine or in grape yield, and she's using field data which were acquired in another project. She's in the meantime able to eliminate those parts which are not of interest for her, namely the leaves, to have a good look on the grapes itself. And by this, she develops this understanding of really what is needed to remove the occluding parts, namely the leaf, to have then really a better view and quantitative view on the parts, namely the berries itself, for a yield prediction. And last but not least, third partner project headed by Diana Pavlik, and she is using different UAV imagery with different viewing angles, separating there in the data the ratio or the fraction between plant and soil pixels, which is a relevant trait for fractional cover. And by doing so, fractional cover is something which is the basis for then really to get a better quantitative understanding of other structural, canopy structural related traits. As I said, only really briefly, I'm looking a little bit on the time. All these projects which we try to highlight and give you a flavor on where we are working, and I hope we got an idea, could give you an idea how they are interlinked. A lot of these projects come together in our central field experiment at Campus Klein-Altendorf, which in past vegetation period, despite Corona, grew a lot. So we are really happy to not only have our central experiment, which you can see here from an aerial view, but that also other experiments moved close by to our central experiment to exploit the synergies between sensors, which then can easily move from the one field to the others, and by combining data, measurement concepts, and also discussions. So many thanks also again to the crew at Campus Klein-Altendorf who made this possible, that we have in the meantime this pretty impressive field design at Campus Klein-Altendorf. And to give you an idea of where we are at the moment, so Campus Klein-Altendorf, we have those fields at Campus North. We have completed the experiment in 2021 South, and we are preparing for 2022. Again, we have and we will install several environmental stations across the fields to have a good coverage of the environmental conditions in our field, not only on one point, but also to cover the spatial heterogeneity. We are still talking about high-tech experimental sites. So you have electricity, you have Wi-Fi, and as I said before, also the synergies to other synergistic experiments, for example, like the breed phase, which is located in between. We are still maintaining the crop rotation, which is important to be close to the practical usage. And 2021, we had our striping field experiment here in the south, and we are now moving to the north. So if you're at Campus Klein-Altendorf, you will find the newly sown fields now in the northern part. And with this, and some impressions from last year's field experiment at Campus Klein-Altendorf, I think I and Sven, I think we are at the end, and I hope that I could give you some idea how the different PhD projects, how they are actually really interlinked in this different 3 pillars of focus areas. And with this, I think I'm at the end of my presentation, or at the end of our presentation, and I thank you for your attention, and we are open for any questions.